Officer for the Central Florida Zoo and Botanical Gardens. I'm also the director. I'm also the director for the Central Florida Zoo's Orianne Center for Indigo Conservation. That's quite a mouthful, so often we'll just refer to that as OCIC. So if you hear me say that, that's just the abbreviation for what I just mentioned. I'm going to go over the eastern indigo snake with you. Just talk about some of the natural history, some of the things we're doing for conservation, and since I'm a veterinarian, I'll talk about some of the health things right at the end. I'll squeeze that in. This is this not going to work? That's what So it's very appropriate. This is the year of the snake. If you follow the Chinese New Year, so you want to go ahead and go forward. A lot of fun to do was lost, but we're going to talk about eastern indigo snake. And so the scientific name for eastern indigo snake is Drymarchon couperi. And that roughly translates into emperor of the forest. Emperor of the forest. Yes. So what are they? So they're this large-bodied diurnal colubrid. So they're a daytime snake. And they're the largest, the longest snake native to North America. You see here the record length is 103.5 inches. That's 8 feet, 7.5 inches. That's a big snake. But most of the boys are between 6 and 8 feet. Most of the girls are between 4 and 6 feet. Overall, they're this bluish black. They're so blue, they're black. When the sun hits them just right, they have this really cool iridescent sheen. They've got this creamy white to an orange color on their chin. And sometimes it's just a small little spot like a goatee, or it can be maybe a third of the way down their body. They have these big, smooth scales. The boys, when they hit sexual maturity, the vast majority of them will get these little keels. You can see here these little raised areas. On that center, on dorsal midline, the center 1 to 5 scales. We do have a couple girls that have these as well, so it's not 100%. So I think of it as like the mustache of snakes. Sometimes both have that. And they're listed as federally threatened species since 1978 by the Endangered Species Act. So they have this huge home range. One snake will have a large territory. So up in Georgia, there were studies that had shown that the females are claiming about 800 acres, one snake, and the boys are 3,000 acres. Now the snakes that live in Florida tend not to take such a big territory. The grocery stores are closer, so they don't have to go so far. But to give you an idea, 640 acres is 1 square mile. So these boys, 6 square miles, that's a lot growing on your belly, right? In fact, one snake in Georgia was captured and then recaptured 2 years later, almost 15 miles away. Historically, they lived, you can see that little map there, the entire state of Florida, you'd be able to find indigos. The southern third of Georgia, and the little dangly parts of Alabama and Mississippi. Currently, you're only going to see them in that dark green area. And this map is a bit outdated. That green area is more splotchy now. They will live in a wide range of habitats. And you can imagine, everything from Georgia all the way down to the Keys, the habitats, the state is different. So They'll have all those. They like to, up in the northern range, they like to, in the summertime, get down in the swampy areas. That's where all the groceries are. But in the wintertime, they'll come up to the high sandy hills and they'll find gopher tortoises, and they like to get up into the gopher tortoise burrows so they can stay warmer because it gets really cold, from a snake's point of view. We see a wide variety of habitats. The longleaf pine ecosystem, which is a photo here of longleaf pine forest, that's one of the most imperiled ecosystems on the planet. We hear about the Serengeti, you hear about the Amazon rainforest. The longleaf pine used to be over 93 million acres in the southeast part of the United States. We're now down to less than 3%. So we mentioned the gopher tortoise. So the gopher tortoise is an inhabitant of this longleaf pine ecosystem. And they're considered a keystone species. A lot of other species rely on the gopher tortoise. Why is that? They're a gopher tortoise, right? They're in a little burrow. So these burrows they dig, they'll dig them down a good, they can be 20 feet down and sometimes 20, 30 feet in. And we're keystone because a lot of other critters will use this burrow, right? So the gopher tortoise dung beetle is only found inside gopher tortoise burrows. We have gopher crickets, cave crickets, primarily use gopher tortoise burrows. We'll see gopher frogs will primarily use gopher tortoise burrows. The Florida mouse, six-line race runner. These guys all inhabit these burrows. Ooh, yeah. So diamondback rattlesnake. Where's Fred? So all these guys live in there, and our good friend, the eastern indigo snake. So why would they use these burrows? We said that we primarily for thermal regulation. We stay warmer in the winter. And you would think with all the other things going in there, hunting ground. That's a good place to get some groceries in the winter. They also use it for protection against fire. For this longleaf pine ecosystem, regular fire is needed for it to stay healthy. And before Europeans settled the area, lightning strikes and the woods would catch on fire on a regular basis. And it helps keep the underbrush from becoming too thick, and that grassy plain, that grassy savanna, is really just right to allow the tortoises to walk around. So they'll get down in those burrows to protect, get away from the fire. And there was a publication that had demonstrated that this snake was succumbed to a fire because it didn't have a burrow. Now they may share the burrow with a tortoise, but quite often they'll use an abandoned burrow for their tortoise. And it's been shown that they come back to the same burrow year after year. So they may go out in the summertime into the swamps to go do their summertime things, but in the winter they come back to the same burrow. So they have site fidelity. In regions where there's less gopher tortoises, they'll use other things. They'll use stump hollows, they'll use armadillo burrows, even seem to use land crab burrows. And here's a stump on a tree, there's indigo there, it's kind of dark, I'll lighten it up, you can kind of see them there. I'll throw it up in there, so it's just kind of nestled in. Okay. What will they eat? Here's some pictures of things that have been found inside the stomachs of eastern indigo snakes. They'll eat anything they can overpower. A shark, yeah. They've been known to eat sharks. There was a paper published in 2014 that just had listed all the things that were found inside eastern indigo snakes, just a small little list. And pretty much they found everything from snails to bugs to amphibians, frogs, salamanders, turtles, tortoises, rodents, alligators, fish, birds, mammals. Pretty much anything they'll fit in their mouth, anything they can overpower, they're going to eat it. They are not venomous. They're not a constrictor. They're just big. We said they're almost 8 feet long. They're just a big, strong snake. Now, all those things they eat, the favorite thing they like to eat are other snakes. And the next picture is going to be a little graphic. Their favorite thing, their favorite flavor of snake are the spicy noodles. They like to eat venomous snakes. So they like the copperheads. You can see here the photo on the left, eating a copperhead. And they'll eat diamondback rattlesnakes, photo on the right. They're immune to their venom. Pretty cool, right? This snake is awesome, yeah. So we call the eastern indigo snake a lynchpin species because they help control the population of many other species. Keep their populations in check and balance. But they tend to be calm. Cramp in my foot, sorry. When they're handled by a human, they tend to be actually pretty calm. They do have some defensive behaviors. So some things that you'll see to be defensive, they might have neck flaring. You know, like a cobra will hood up, and you'll see the side to side puff out. Indigos will puff their throat out and turn their head sideways to make themselves look bigger. They'll hiss. I call it talking. They kind of talk to you, but a lot of times when they're not wanting to be handled, like they see Nick come in, they're going to go ahead and give him some grief and just kind of talk like that. How was that? Is that okay?
That sounds just like
I'll give you a 20 later. And death feigning. So this is not widely talked about, but there have been signs, or they've been found to play dead. And this picture was taken, Dirk. This is one of the Dirk Stevenson's photos, a biologist up in Georgia. He had seen an indigo, he thought it was dead, and took a photo of it to document it, and then it started moving. He's like, oh, you played dead like a hognose snake. So they don't do it a lot, but it's been documented. What are their threats? Habitat loss. It's kind of hard to find 6 square miles of anything anymore, right? Habitat loss, fragmentation, got a road going through it. So here's the longleaf pine, what we should be striving for, where they want to live. We got degradation of that, so you can see just overgrowth with its fire suppression. So if that's burned regular, you get more of that previous habitat. Gopher tortoise population declines. So as the gopher tortoise goes down in numbers, then there's fewer homes for them to live in, especially in the northern latitudes of their range. It's a big, there's a big black snake. Let's kill it. Yeah. That's one of their threats. And collection for the pet trade. So that's been put to rest. That's not allowed to be done anymore, but prior to 1978, that was a common thing. So what can we do? So the Association of Zoos and Aquariums has multiple safe programs, saving animals from extinction. The eastern indigo snake is one such animal in these zoos that have a safety program. And here's a handful of zoos across the country that support the eastern indigo safety program. And so they're doing education and bringing awareness to the plight of eastern indigo snake. More locally, we have the breeding and reproduction program. And that is primarily centered at the Central Florida Zoo and the OCIC. And we have several key partners that are listed up here to help us do this. And we're breeding the snakes in captivity under human care for the sole purpose of releasing the offspring back into regions where their numbers are extirpated or that are locally extinct. Here's the OCIC. So we've got several buildings. Historically, eastern indigo snakes were tricky to breed in human care. It was hard to have good success. And our first director, Fred, said, if they're native to this area, why don't we keep them outside? Maybe there's something that's in the environment that helps them do better breeding. exposure to the different barometric pressures, the weather patterns, the change in the day and night cycle, the length of the day, length of time of night. And so these outdoor habitats were constructed. There's a photo there on the right of the outdoor habitat. And we had good success breeding outside. Now, in his great wisdom, he also said, well, we're going to build the herpetarium. We have snakes, may not have enough bedrooms outside for them. We want to keep them inside too. Let's make sure there's windows. How many herpetariums or serpentariums you go around have windows? Not a lot. They don't have it. So you come in, you work an 8-hour day or a 10-hour day, and that day and night cycle is always 8 hours, 16, 8, 16. And these windows, you work your 8-hour day and you leave, you still have the natural light coming in, and the length of day changes as the season changes, and we've had good success breeding inside as well. Thank you, Fred. So these outdoor habitats are pretty cool. So we have, each snake has its own bedroom. They live by themselves because they eat other snakes. We try to set it up as naturalistic as possible. So we have these really cool habitats. We put the plants in there, and we try to simulate a gopher tortoise burrow, right? You can see, let me try this. Yeah, all right. So right there is a cooler that we countersunk in the ground. We drilled a hole in the side and made a little tube. Gopher tortoise burrow. And if they're down in there and we want to check them out, it's got a lid. We can look and make sure they're okay. So this is a nice setup here. And we can see here we have like this little structure and this structure. So we've taken the tube and we made it convertible as well with these plastic, it's hinged. And so they're not known to be arboreal, but we find them in the branches quite often and climb up in the tree. Each snake has their own personality. Like 417, he's always in the branches. 152, she's never in the branches. So we number them as we acquire them, so they're affectionately, 417 and 142. So we can see here, oh, look at that pretty snake. Looks like a couple of minutes there, just enjoy it. And, oh, there's that little, off that side there, we lift that lid up, and we can see down inside our little tunnel going in there. I like to utilize that as well. That's pretty cool. So currently we have 163 snakes at the OCIC. We have 38.28 breeders. What does that mean? 38 boys, 28 girls. That's kind of how you say that in zoo talk. We've got 25 yearlings, and we've got 42 hatchlings. Actually, these hatchlings just left last week, and we'll get to that in a minute. So that's our current population. And we've got 97 eggs incubated. So we're not counting those guys yet. One thing that's really interesting about these guys is they breed in the wintertime. Most snakes breed in the summer or in the spring. They're backwards, they breed in the winter. Because they're coming out of the swamp, going up to the sand hills, and that's where they're going to find other indigo snakes. So that's a great opportunity. And so what we often do is we'll have male combat. Has anyone heard about male combat? Know about that with snakes? No. So two boys will duke it out, and the female then will pick the winner, or she will decide to breed with the winner. And so it's kind of funny if you ever see it. There's not usually bloodshed, but it reminds me a lot in grade school when you used to do thumb wrestling, right? And, these two snakes, you see them one on top of another, kind of, I'm going to win, I'm going to pin you, and it gets pretty heated, and they drool on each other's face, and then they shove their face in the sand and try to sand. So
No, they're not. Very quick.
Great. So every once in a while, there can be some injuries. So we don't, hey, that will help get the testosterone going, and that might be better for breeding. But we found over the years that that's not necessary. We don't have to do that to get successful breeding. And we have had a couple male-on-male injuries, and we have to supervise the event. We have to supervise breeding because we don't want one to eat another one. That defeats the purpose. So date night is always chaperoned. And then the fight night would have to be chaperoned as well. We spend we spend over 300 hours in just chaperoning. Why take the extra time and chaperone the boys combating or breeding if we don't have to make success? So we've stopped doing that. We really haven't done that for a few years. But you can see up here, these two boys here are thumb wrestling, and she's waiting to pick for the winner. Oh, look, there's Nick in the winner. Then they lay their eggs in the spring. So we're just now coming out of egg laying season. We have one more female that we're waiting on. And they'll lay anywhere between 5 to 14 eggs. And they're big. So you see they're pretty good sized eggs. We then will collect the eggs and we'll artificially incubate them. And we'll incubate them for about 100 days. And in the summer, the babies hatch. Oh my God, they're so cute. That's summertime. So that's a few months away. We'll hang on to them for a year. And then when they're just about a year old, we send them off to one of our Head Start partners. And that's why I said, we have these hatchlings that, we just lost them last week. We sent them off to one of our Head Start partners. And they just take care of them. One of the Head Start partners is Zoo Atlanta, and our other partner is the Willaca National Fish Hatchery. They have a great conservation program there. But it just spreads the cost. Spreads the love that someone else gets to feed these guys and clean up after them for a year. Once they're 2 years old, that's when we're planning on releasing them. And where are we going to release them? Currently, there's only 2 places we're releasing these guys. There's a lot of criteria involved for releasing the indigo snake. There has to be an area where they've been shown to not be for several decades. Makes sense, we're going to release them when they're not, but where they should be, where they historically have been. The area has to have several thousand acres of restored longleaf pine forest that undergoes regular fire management. There has to be X number of gopher tortoises per acre. And so it has to be non-fragmented by roads, or at least minimally fragmented by roads, and preferential treatment goes to sites that abut against other tracts of land that are similar. So our one site is the Conecuh National Forest in southern Alabama. That fits the bill. Indigos haven't been seen there since the 1950s. And the other site is in the Nature Conservancy's Apalachicola Bluffs and Ravines Preserve, right up against the Torreya State Forest and the Apalachicola National Forest. So those two fit the bill. And indigos haven't been seen there since 81. So that's like 45 years. I can't believe 81's 45 years. So that's what we got. Delay here. Clicking. Yeah. So we transport the snakes individually in our little containers there. We take photos before we release them, get their mug shots. Take a photo from the top, photo from the right side, left side, and the bottom. Each snake has unique physical characteristics. Out to an indigo or a gopher tortoise burrow, and we release them into the gopher tortoise burrow at one of these sites. So taking photos, here's an idea, like, oh yeah, the snakes look the same. But you really look close, we can see, like these scales here aren't the exact same as those scales there. Kind of like a fingerprint. Similar but different enough. And so we can help identify snakes by their physical characteristics. We also put microchips in them, so that helps too. But you know, it's fun taking a picture, we do that. To date, we've released 327 indigos in the Connecticut National Forest, 240 which were from OCIC. The other ones were released there prior to our involvement, and 209 at the Nature Conservancy's Ecological House, the Panhandle. Do they have trouble adapting? Born in captivity, been under human care their entire life for 2 years. This next little slide is going to be a little graphic. There was an indigo snake that was released. Within 1 week of being released, found, caught, killed a copperhead. Never saw a copperhead before in his life. This was a little video taken by one of the master's students from Auburn who was doing some of our monitoring early on in the project. And that's pretty amazing.
They just swallowed the whole thing?
Yeah. Right on. Right here, we don't know what happened to that one. Survival estimates, the first year we were monitoring them, there was 12 indigos released that year. Just under half survived. The second year, 20 were released, just under half survived. So about half are making it, not quite half are making it. And we don't know if these numbers are skewed. This is sampling bias, get into research and that kind of stuff. We're only finding those that have radio transmitters in them. Are the radio transmitters causing something to allow them to not behave quite normally and allow them to die easier? We don't know. So that was only done the first couple of years, but we don't do that currently. What do we do then, since we're not doing these telemetry surveys with a transmitter? Pedestrian survey. Walk around the woods and looking for them. Our biologists will walk several miles. I think Michelle tells me on a easy day, she walks 9 miles every day. So she thinks she gets paid by the mile or something. So we look for signs of snake activity. So gopher tortoise burrow, you can kind of see here, you can tell a snake was crawling in and out. It's called a snake crawl. Right? That's one thing. Oh, we know there's a snake here, and then it can hedge our bets. Other days where you're going to start looking. Or a dirt road, you might see a snake crawl going across the road. Hey, you find a shed skin. There you go. There's a snake around here. That's one way of telling. We are able now to take a sample of this shed skin, and we've got PhD postdoc working on the genome of the eastern indigo snake, and he could tell us the genetics from that shed skin, who the snake was. If it wasn't all crinkled up like that, and you catch a snake. Hey, that's one bite. Look at there, I found a snake. But if that shed wasn't all crinkled up, you had just the skin of that shed, we could go back to our photo library and figure out who that snake was, most of the time. Really interesting, cool news. January 2020, a hatchling indigo snake was found in the Connecticut National Forest. There have not been indigos seen there since the 50s. We released 2-year-olds. They're sexually mature around 4 years old. So that means they're surviving, finding each other, and making babies. Yeah, pretty cool. And in 2023, on 2 different occasions, in the ABVP and the Panhandle, same thing. Yeah. Feel like we're doing something. Other things that we do to look for snakes, we'll set up these drift fences, and we'll set up camera traps. So we started originally, we took a camera, like a field camera, like we put out for deer or whatever. And the problem is, if you have it set for motion, in order to pick up a snake, you have to set it very sensitive. And then it picks up every blade of grass that's moving. And so you get all these photos that have nothing in them. So instead we do time lapse. That's like a photo taken every 30 seconds. Well, that's a lot of photos too with nothing in them. but it's less than the blade grass photos. We then put in these drift fence arrays. And so it's basically a silk fence like around the construction site. They put it so the debris doesn't flow in the road. So around the middle of the woods, we make this like big X. And at the center, we put a bucket upside down, and inside that bucket, we put a camera trap. And we can set it on motion then, because the only thing that's going to pick up is what's going underneath the bucket. We've got millions, literally millions of photos from both the time lapse and these bucket traps. And that helps us find out what's out there when we're not actually able to walk around. Here's one in front of a gopher tortoise burrow picking up on time lapse. A gopher tortoise, imagine that, in a gopher tortoise burrow. Well, that's pretty cool to see that. We've seen indigos too. That's cool. Sorry. There it was.
Just one.
But they also, the camera traps, the ones that go under the bucket, we've picked up, of course, the snakes, lizards, tortoises, mice, other animals, birds, bugs. And you can see here, there's a Florida mouse.
Oops.
Sorry, birds coming popping through there. Everybody here likes birds. And indigo snakes. Yeah, that's pretty cool. You can see, with the different patterns on the top of the head. We just now started to get some technology involved. So we've got, we've been going through these photos, and Michelle will go through, our field biologist will go through these photos and line them up with our mug shots and look at the different patterns on the back of the head. And oh, that was 384. All right, I mean, she's really good at it. I can't do it, she can do it. But we've now started getting these automatic pit tag readers, passive integrated transponder. That's just another word for microchips. We get these microchip readers, and we're putting out there this big 4-car batteries hooked up to this big cable that we'll put it around the camera trap. So that way, if we get a photo of an indigo snake, timestamp, we go back into the log here and find out, was there a microchip associated with it? That was snake 432. Great, Or if we get a photo of an indigo and there's no microchip associated with it, that's a new one. That may be one of those babies that have made it on that we didn't catch up before. We are now hooking those up to solar panels, so we're going to keep going back out there trying to recharge these car batteries out 2 miles in the middle of the woods. In the summertime, we'll set these drift fence arrays kind of like that line here on the left. Every 100 feet, there'll be a bucket, and then we might have a camera only at, we'll have the camera at each bucket, but we'll only have the microchip reader at certain ones because we can only afford so many of them. And then in the wintertime, we do the big X. And then the camera traps on the ends, and one in the middle's got the camera trap and the microchip reader. Other projects that we've got going on looking at monitoring, environmental DNA. Has anybody heard of eDNA before? Yes, one. So this is really cool. This is something that's been done a lot in aquatic species, where they'll take a sample of water and they could check that water and see if there's hellbender DNA. And then somewhere upstream, there's the endangered species hellbender, or there's several different fish that will do the same thing, or endangered species of crayfish. They can find the DNA in the water and they know there's something in there. So we thought, what if we could do that on land? So let's look into that. We took, we were working with actually the Department of Defense of all people. We're looking at trying to see if we can detect eastern indigo snake DNA in sand. And so before we start looking in the wild, let's see if it can be done. So at our center, we set up these habitats that never had a snake in them. And we would put a snake inside this quadrant, then sit there for a minute, take it out. And the next one, set it for an hour, take it out, 2 hours, take it out, 4 hours, take it out. Then we would take a sample of the soil, 6 hours, 12 hours, 1 day, 2 days, 3 days, 4 days, 5 days, all the way up to 10 days. And there's Rhiannon taking a sample of the soil. And we found that you can, if the indigo's been there at least an hour, we can get DNA for up to a week. That's pretty cool. And these are outside, so it's an exposure to the just natural elements, sunlight and rain and all that jazz. And our experiment, in the middle of the experiment, hurricane, whatever it was, there in August of 2023, that came through. Made us miss a couple sampling times because of the hurricane, but so even through that, we were able to still pick up DNA. And we are currently doing some field studies where we're getting samples from the apron of the gopher tortoise burrows, and actually it just got accepted for publication in a peer-reviewed journal, that we are finding it in the wild as well, and if paired with pedestrian surveys is one of the better methods, superior to drone or cameras, actually. So that's a pretty neat technique that we're going to start expanding. What else do I have coming up? Oh yeah, using the photo matching techniques, which I was alluding to earlier. So, here's snake 511, and you say, oh, look there, yeah, see those scales. They all look the same to me, right? But Michelle found this shed skin, and looky there, was able, it took her a while, going through all the photos, and finding that little pattern, she was able to tell that shed skin was from 511. That's pretty cool. How can you help? If you are so lucky to see an eastern indigo snake in the wild, go to the FWC webpage, rare snake sightings. There's a link right there for eastern indigo snake. Click, and you can put in GPS coordinates. It just helps us determine where they are and where they aren't. You can also help us through Zooniverse. So this is a citizen science project. I said we got millions of photos. We need help. We upload these photos. Currently, the person who was uploading them was employed by FWC, and something happened in the government. So the things have not been uploaded for a while. This summer, it's going to get back up again. We can only upload 10,000 photos at a time. Once they get gone through, then they can come off. So there's a tutorial on here. You get on there. You can help us go through photos. You don't have to, worry about how I'm going to miss something or whatever. There has to be an agreement between 10 people before that photo gets retired. And so you really help us go through millions of photos. The photos that we're catching that are being uploaded, quite often we're seeing nothing. But we can see some of the photos that I showed you earlier. That is a QR code that links you straight to Zooniverse. If you're interested, you can go right there. Again, there's no photos right now because they've all gone through and we're waiting for uploading. And I actually have one of my field techs. That's his mission this summer to start loading those back up. So that'll be available soon. Like I said, I'm a veterinarian, so I got to do some health things. So there was a survey a few years back with all the AZAs, what's seen in indigo snakes and under human care. And when they get older, they get cataracts. Quite common. You can see the cataracts in that gal there. Sometimes they get retained spectacles. So when they have their skin and they shed it, sometimes the part that covers their eye doesn't come off and gets stuck. We just have to remove that. They get skin lesions. Some of these can be pretty bad skin infections. Some of those skin infections can get septic. The boys can sometimes get their boy parts infected. There's a horrible parasite called cryptosporidiosis, or a parasite called Cryptosporidium serpentis, and the disease is cryptosporidiosis. And sometimes the eggs get stuck, dystocia, and we have to do a C-section. There's a surgery, intraoperative photo there of a C-section in an indigo snake. Thank you very much. I'm happy to answer any questions.
Can I ask
Makaiva, have we ever introduced snakes there and why? Right, is that what you said? Makaiwa?
Did I miss when you talked about that?
Yeah, go, watch the tape. Yeah, so we have not done anything with Kaiwa. There has to, it's very strict on where we're releasing it. We're only releasing at 2 sites, and we're not expanding to a 3rd site until we can demonstrate that we've got success or getting towards success, and we're getting there with those babies that we've seen. But we just had a strategic decision workshop last month with all the major players, and we came up with some parameters to help determine when we're getting successful. Early on, back at the napkin type of math, we said, you're going to release 300 in 10 years. Numbers pulled out of someone's ear. And so, we actually went through some math and trying to figure out what we would need, like, oh, we want to make sure we not only see babies, but we want to see babies of babies, and we want to make sure that we're still seeing goat tortoises, and we make sure that they are expanding outside we're releasing them, we're finding them, within a few miles of there. So there's a whole bunch of things we're looking at. If they have to have
14 acres is what they have.
So that is, that's a bit small. So the ABRP has 8,000 acres and abuts against 2 national forests, which have multitude more of that. The Connecticut National Forest has 15,000 acres, 12,000 acres? Yeah, a lot more. Our 3rd site that we're considering is also in the Panhandle. Indigos have not been seen there for over 40 years, and they've got 175 acres of longleaf pine forest. So, and tons of goat horses. So they'd be the next one if we expand beyond that. So there has to be more to it than, I got a big yard,
Are you finding snakes in the Wakaiva area these days, or?
So that is not, we don't do the field work in the Central Florida area, or we just monitor where we release. And most of the other monitoring throughout the state is done by FWC. We're working to expand into those regions as far as to help out, but right now, I couldn't tell you.
Question?
Yes, sir.
Well, that's Fred, but I'm right behind you. So a couple questions. So captive snakes, how long do they live as opposed to, well, released snakes could die quicker, I realize, but, you have enough snakes right now, or are you still collecting more snakes? So you got enough for the breeding program right now? And then what were you saying also about you weren't putting chips in them anymore because you thought there may be problems, or what were you saying about that?
So a multi-part question.
Yeah, right.
Yeah, I lost the first part there.
Captive snakes. And
So how long do snakes live under human care? So the average is in the mid-teens, although there have been snakes documented in AZA zoos that have lived to be up to 30. We just had, in Eastern Indigo snakes, we just had a 15-year-old female lay 6 eggs yesterday. So she's going to keep going, right?
Yeah.
What was your other part of that?
Well, I was saying that snakes released and whatever, but that's probably going to be more
Yeah.
short-lived.
Yeah, so when they're released, we don't know yet,
Yeah. And you're
they're
still-
they're very hard to find out when they get released. Put out
Yeah,
there.
right.
There's a snake that we thought was lost, that was at the Connecticut in the Alabama area, that was just caught again this past winter, and she hadn't been seen for 5 years. So it's hard to tell, right? And then as far as the implants, we still microchip all of them. The radio telemetry unit, radio transmitter. So a microchip is a passive integrated transponder. It's not a low jack, right? So if it goes, you put it in there, you can find it, boop, you can ID. But you can't get on your phone, let me go find them. It doesn't do that. But if you have, one of these things, surgically implant them into the snake, then we can get a little machine, like a Geiger counter type of thing. And when the beeps get closer together, we're getting closer to the snake, and then we can track them down. But this is pretty big, and this has a battery in it, and it only lasts 2 years. So we do have to make sure we catch them up again, find them, so we can surgically remove it. I don't know, is it causing a problem? This is inside, I mean, this is like a, I don't know, fire extinguisher we put into you, right? Might cause some problems if it hooks up in there.
You quit doing that? You're saying one of those you quit?
Yeah, so we don't do this anymore.
You don't do that.
So we do that the first 2 years at the,
Okay.
at a new release site, which we're only 2 release sites. And that way we get a rough idea where we're gonna start looking. So we know, oh, they tend to head this way. And then future years, that's where we're walking and looking, is that way.
Where do you get most of your breeding stock, or all of your breeding stock? You rely on people breaking it in, or?
So our founders were collected in a stable population in Georgia. There were gravid females or pregnant moms were collected, brought to Auburn University. They laid their eggs there. And the majority of the babies, while the moms went back to Georgia, and the majority of those babies were released, and that was part of those original numbers that we talked about. And then 25 or so, about 25, 30 snakes from there, to come down to OCIC, and they were our founders. They were our starting bunch. And then through the years, there have been a handful of snakes that were collected from by FWC that, for whatever reason, their habitat was no longer suitable for the indigo. Strip mine, something was going on, and so they were then collected and donated to us to increase the genetic diversity of our breeding stock. Yeah. Brad had a question.
Yeah, so the eDNA, I know that the source here is
It's
indigo
smelling in front of
snake.
us.
But what is it fecal material, is it oil? What is the DNA source from the indigo?
Yeah, their essence, evidently. So what's the source of the eDNA? But it doesn't make sense. I don't know. They're just called something on their, because obviously they're not shedding skin cells like we are. They shed a whole big thing at once. But just the fact they lay in that was why we did the study to see. And yeah, the DNA is just leaching out of them. I don't know if it's through the respiration and droplets settling. I don't know. I don't know that we know what that DNA
Yeah, it's
is.
hard to get your head around that.
Yeah, it's kind of weird.
Really
But-
neat.
Yes, you had a question.
Yeah, the longleaf pine forest you release the snakes into, are they working forests? I mean, is there any harvesting of the trees, or it's just wild?
Wild.
Okay.
Yeah.
But they do have prescribed burns then?
They do prescribed burns, that's right. So depending on the site, anywhere from 2 to 3 years, they're burning that, and that keeps all those invasive plants out, it keeps the shrubs, the bush from coming up, and it helps keep it that grassy savanna that allows for all the animals to move around better. Yeah. Yes, sir.
I don't know, Deb was over there, I've seen an indigo in the wild at Wakanda Spring State Park years ago.
Yes.
And my question is, a friend has told me that pygmy rattlesnakes in particular are dying because of some kind of lung infection.
Right.
Is that true? And is this affecting all snakes or indigo snakes?
Yeah, so, boy, can I talk about that. So, go, veterinary, all right. Yeah, so there's this parasite, a lungworm, called a pentastome, right? And they're creepy little things. They're not really worms, they're crustaceans. They look like worms, but they're actually crustacean. And they parasitize the lungs of carnivorous reptiles. And Chirocephalus is one of the lungworms that's native to the region, and snakes may have 2 or 3 of them and whatnot, and they just deal with it and they're fine. Reptile orientalis is an invasive pentastome lungworm that came along with the Burmese python. And the Burmese python invaded and became an invasive species in Florida. Their parasites decided to spread the love and go to native snakes as well. And so you can imagine a parasite for a giant snake getting into a little tiny snake can be devastating. And they seem to go hog wild. So where a Chirocephalus or a native pentastome a snake may have, 2 or 3 and can deal with it. These pygmy rattlesnakes, or a lot of the rodeo water snakes, are getting pentastomes from the Burmese pythons that are just going banana sandwich. They might have 30 or 40 parasites in there. And then they're big enough on these little snakes, we're gonna try to crawl up, there's no room in here, and they occlude the trachea and choke them out. And we have found them in wild indigo snakes. So yeah, they're a potential problem, and they're moving farther and farther north. And not the Burmese aren't, but the worms are. They're coming up through some of the native species. Yeah, good question. You had a question.
I'm sorry.
Oh, another multi-part, okay.
Yes, but I'm going to do it one at a time. So one of your first pictures, you had 2 heads, and one looked like it had a red nose. Is that male, female, or just a coloration, or age?
Just a variant.
Okay.
Just, some people have red hair, some people have black hair. That's
Okay.
that.
The other thing, once the babies they look like they're both all put in an individual drawer.
Correct.
And is that because snakes, that indigo will eat an indigo?
Yes.
Okay, and then what do you feed them and how many times a day while they're in the drawer?
Good question. So what do we feed them? So we feed them what's easily acquired, right? We feed them, if you're in the hobby and you have a pet snake, you can buy frozen mice or rats or whatever, depending on the size. So we'll feed them that because it's easier than trying to have a whole colony of mice to feed them. So we have frozen items, and we'll rotate between mouse or rodent, bird. We'll do chicken chicks or quail chicks. We'll feed them fish, either capelin or trout or smelt. We'll feed them frog legs. So try to rotate, give some diversity. I'd love to feed them other snakes. I have 163 snakes, and they eat twice a week. So I would have to have 330 snakes to feed these snakes every week. Where am I going to get that? So we're actually, so some of the things like the dystocia or the thought is that it may have something to do with fitness. They're in human care. Even we have the outdoor unit, a big 10 by 10 bedroom. It's not 200 acres, right? So, and we're feeding them these thawed frozen lab rats. In the wild, they're eating snakes. And they're crawling, hundreds of acres. A snake, if you run a snake through a machine, a snake's a food item. Snake is high in protein, low in fat, high in ash. Ash is that non-digest roughage. Everyone would say roughage. So that's what a snake is. So that's what they're designed to eat. We're feeding them lab rats, which is moderate in protein, high in fat, low in ash. So instead of having this Olympic athlete eating a keto diet, we've got these couch potatoes eating cheeseburgers. So I think they're probably just not as fit, and they're having problems laying eggs. So we're actually looking on trying to get a diet that's more similar. We've done a year-long study making a sausage that has the same nutrient profile as a snake. And yeah, it's worked out over that first year. Yep, their blood values are looking good. So now we're going to do a multi-year study to see if it'll help decrease the incidence of their egg binding issues.
One more thing.
One more.
Someone was telling me the other day if they'd seen a gopher tortoise in our neighborhood. If you see a gopher tortoise, would you all like to have it? I mean,
No. No.
Just checking.
Yeah, you see gopher tortoise do a little happy dance. You got nature with you.
No, well, we used to have fox turtles, and then we acquired some gopher tortoise, so we have stories on gophers.
Yeah, nice.
They're fun, yeah.
Yes, sir.
Couple of questions.
multi-part questions, dang.
How many indigo snakes do you see in a camera trap? Just the same individual over time, or do you see multiple individuals using the same area?
Both. So we'll see the same one over and over again, and we'll see multiples, and we have hundreds of thousands to over 1 million photos still to go through, so we might be able to answer that better if we get, help us out, look through that, pictures there. That'll really peel that onion back more.
And what about genetic diversity in the entire population? Is it pretty low because of the habitat loss, or?
So the genetic diversity is a good question. I had some preliminary results from that doctoral student at Auburn, and the genetic diversity is pretty good through the peninsula. Out towards the periphery, it's not as diverse, but that is pretty typical for species. When you get towards the edge of a range, obviously you can only interact one direction. You can't interact that way. So normal for the way species normally act, the genetic diversity is less on the periphery of the range. So not too bad, actually. So there's hope.
Good.
Yeah. Yes, sir.
Commercial time. 3 years ago, I went to the Central Florida Zoo website and saw the OCIS link there, and I visited the facility out there in Lake County with Nick. Are you still doing that?
So the OCIC, commercial time, so are we still doing tours of OCIC? We are and we aren't. So we were trying to do access to the public for scheduled tours, but our facility is not ADA compliant, and it's not really conducive to do that. So there's a lot of infrastructure we need to go in for us to do it right. So we're burning money elsewhere, so we're not doing that. But we do have tours for conservation and for school groups, conservation groups.
Okay,
So we'll have
okay.
intermittent tours by appointment.
Yeah, because when I did it with Nick, I got my crutches out and got on the cart, and then we went to the center there and the old habitat.
Mm-hmm.
Okay, and
You said Nick, so I figured.
yeah, okay, yeah. So I mean, I learned a lot on that day, and I still remember it. And he's never been bitten by a snake. So I mean, anyway, okay.
Yeah.
I
Yes.
don't know if you've seen it or not, but your last release, you had a lot of press coverage on them. I've seen the story on several news stations.
Yeah, it's exciting.
This week, so you've had, that's good.
Yeah, thank you.
Yeah.
Well, I think people would love to see this